Welcome to our exploration of the X chromosome, focusing on dosage compensation, X inactivation, and their clinical implications. By the end of this lecture, you'll be equipped to define dosage compensation, explain the N1 rule, describe the Lyon hypothesis, discuss clinical implications, and identify genetic bases of Fragile X syndrome. The X chromosome is crucial for body development, with males having only one X, making them more susceptible to mutations. Females have two, but only one is active to balance gene dosage. Females have two X chromosomes, creating a potential imbalance in gene expression. X inactivation ensures only one X is active, maintaining balance and preventing developmental issues. In cells with two X chromosomes, one is inactivated to prevent excess protein production. This inactivated X becomes a dense structure called a Barr body, visible under a microscope. The discovery of Barr bodies in 1949 revealed the cellular signature of X inactivation, visible in female cells but absent in males, confirming the process in human tissues. Barr bodies are transcriptionally silenced X chromosomes, visible only during interphase, not mitosis, highlighting their role in gene regulation. The N1 rule states that only one X chromosome remains active per cell, with others becoming Barr bodies. This rule helps predict the number of Barr bodies based on X chromosome count. In the 1960s, Mary Lyon proposed that X inactivation is random in each cell, occurring early in development, a principle now known as the Lyon hypothesis. X inactivation is random and permanent, with all daughter cells inheriting the same inactive X, ensuring consistent gene expression across cell generations. Before differentiation, female bodies become mosaics of cells expressing either maternal or paternal X, leading to diverse cell populations. Mosaicism explains varied phenotypes in X-linked heterozygous females, such as calico cats. In humans, it affects disease expression based on which X is active. X inactivation is controlled by the X inactivation center, with the EXIST gene playing a key role. Some genes escape inactivation, explaining certain syndromes. Let's explore the clinical implications of X inactivation, focusing on how it affects genetic disorders and syndromes. In X-linked recessive inheritance, manifesting carriers show disease features due to skewed X inactivation, affecting the expression ratio of normal to mutant alleles. Female carriers of DMD mutations may show muscle weakness due to skewed X inactivation, highlighting the need for careful clinical evaluation. Skewed X inactivation can lead to significant clinical outcomes, such as Rett syndrome, where the degree of skewing affects disease severity. The N1 rule ensures extra X chromosomes are inactivated, but escape genes can still cause phenotypic effects, as seen in Turner syndrome. In Klinefelter syndrome, the second X is inactivated, but escape genes still cause learning difficulties and other symptoms, highlighting the complexity of X inactivation. Fragile X syndrome is a leading cause of inherited intellectual disability, affecting both males and females with significant clinical implications. Fragile X syndrome is caused by a trinucleotide repeat expansion in the FMR1 gene, located on the X chromosome. The repeated sequence is CGG. There are 4 allele categories based on repeat length: normal, intermediate, pre-mutation, and full mutation. Normal alleles have 544 CGG repeats and are stable. Intermediate alleles have 4554 repeats and may be unstable but are not clinically significant. Pre-mutation alleles with 55200 repeats pose a risk for expansion during maternal transmission. Full mutation alleles with over 200 repeats led to hypermethylation of the FMR1 promoter, transcriptional silencing, and loss of FMRP protein. In males with full mutation, Fragile X syndrome leads to moderate to severe intellectual disability, autism spectrum features, speech delay, hyperactivity, and anxiety. Females with full mutation show more variable phenotypes due to X inactivation, with about 50% experiencing mild intellectual disability and learning difficulties. The degree of skewed inactivation of the mutant X determines severity. Pre-mutation carriers are usually cognitively unaffected. Fragile X does not follow classical X-linked inheritance due to repeat instability. Maternal transmission risk involves pre-mutation alleles expanding to full mutations during maternal meiosis, not paternal. Mothers with 55 to 200 repeats risk transmitting a full mutation. Paternal transmission involves pre-mutation fathers transmitting alleles to all daughters, who become obligate carriers, but they do not transmit full mutations. Contraction, not expansion, occurs in male meiosis. Thank you for your attention and interest in learning about Fragile X syndrome.